Greetings student. So today we are going to start a new topic, respiration. So let's start. What is respiration? Respiration is a chemical reaction that breaks down glucose to release energy inside the cell. Now a lot of student confuses respiration with breathing. You have to understand respiration and breathing, these are not the same things. Respiration is a chemical reaction and breathing is a physical process. Respiration occurs in every living organism, whereas breathing occurs only in advanced animals. So why do we need to respire? Because respiration releases energy, and this energy is needed for a lot of physical activities. For example, muscle contraction, making protein molecules and other bigger molecules, cell division, active transport, transmission of nerve impulse, and maintenance of constant body temperature. There are two types of respiration. One is called the aerobic respiration. The other one is called the anaerobic respiration. Aerobic respiration is one which requires oxygen. And from the name you can understand, anaerobic respiration does not require any oxygen. So the main difference between aerobic respiration and anaerobic respiration is that aerobic respiration requires oxygen, anaerobic respiration do not need oxygen. There are some organisms that can respire aerobically, whereas other organisms respire anaerobically. And there are lots of organisms that can respire both aerobically and anaerobically. So, like I have already mentioned, aerobic respiration involves oxygen. Now, since oxygen is involved, it breaks down the glucose more completely. You can see in this reaction. The first one is a word equation for aerobic respiration. Here, the glucose is combining with oxygen and producing carbon dioxide and water. The second equation, this is a chemical equation. Here you can see that the glucose is again reacting with 6 molecules of oxygen, and it is getting broken down to produce 6 molecules of carbon dioxide and 6 molecules of water. So from 1 molecule of glucose we are finally ending up with 12 smaller molecules. So you can easily understand that the glucose molecule has been broken down significantly. Because of this, a lot of energy is produced by aerobic respiration and the glucose is broken down completely. We have to remember another thing, that the end product of aerobic respiration is only carbon dioxide and water. During aerobic respiration, lactic acid and ethanols or alcohols are not produced. The second type of respiration is anaerobic respiration. Anaerobic respiration do not involve any oxygen. Here is an example of anaerobic respiration that occurs in yeast, microorganism and plants. Here you can see that the glucose is being broken down into ethanol and carbon dioxide. In the chemical equation you can see that the glucose is breaking down on its own. There is no oxygen involved here. And the glucose is being converted into ethanol and carbon dioxide. So for every molecule of glucose you get 2 molecules of ethanol and 2 molecules of carbon dioxide. You can easily see the difference between aerobic respiration and anaerobic respiration. In first case, the aerobic respiration, the glucose was broken down into 12 smaller molecules. However, in this case, the glucose is being broken down into 4 smaller molecules. So, because of this, since the glucose is only partially broken down, only a small amount of energy is actually released. And as a byproduct, anaerobic respiration produces either lactic acid or ethanol or alcohol. one thing to remember about aerobic respiration is that aerobic respiration is different in microorganisms and plants and then that of animals. In animals, the glucose is broken down into lactic acids and nothing else. But if we check the anaerobic respiration in case of microorganism and plants, the end products are ethanol and carbon dioxide. So in animal, glucose is directly broken down into lactic acid, whereas in microorganism and plant, anaerobic respiration produces ethanol and carbon dioxide. In both cases, a very small amount of energy is released. Between this one, that is the anaerobic respiration in animals and in anaerobic respiration in plants, you can easily see that anaerobic respiration in plant undergoes more breakdown. The glucose is undergoing more breakdown than that in animals, because here only 2 molecules of lactic acids are produced, and over here 2 molecules of ethanol plus 2 molecules of carbon dioxide is produced. So which one do you think produces more energy? Anaerobic respiration in plants or anaerobic respiration in animal? Obviously, anaerobic respiration in plant produces more energy than anaerobic respiration in animals. You have to remember that plant mostly respires aerobically, but then there are certain conditions in which plant have to respire anaerobically. This is one such example. You can see that this field has been flooded by rainwater, and since the plant is submerged underwater, the plant's root does not have any access to oxygen. At this point, the plants will resort to anaerobic respiration. Now, animals can also undergo anaerobic respiration, but only for a very limited amount of time, say for about 1 minute or 2 minutes. But plants can undergo anaerobic respiration for several days. However, if this water remains here for over a week, then the root cells will start to die. In case of microorganisms, they can perform anaerobic respiration for a very long time. In fact, some microorganisms solely depends upon anaerobic respiration. Coming back to anaerobic respiration in animal, we can see that anaerobic respiration in animal releases lactic acid, and the name suggests that this is an acid. So when the anaerobic respiration is performed in an animal, the lactic acid or lactate starts to build up inside the animal system, and this reduces the pH of the organism. And as the pH changes, it interferes with the enzyme activity. And if this goes on for a long period of time, then all the metabolic reactions of that organism will stop and the organism will die. Because of this, lactic acid cannot be stored inside the body for a long period of time, and the body tries to break down lactic acid as soon as it is produced. Now the question is, when does an organism, especially an animal, performs anaerobic respiration? In our normal day-to-day activity, we breathe in normally. We get a lot of oxygen inside our body. So in normal condition, an animal depends solely on aerobic respiration. But now let's think of a situation where you are heavily exercising. For example, you are climbing up stairs or you are running in the street. When you are extremely active, your oxygen demand rises. Your breathing rate also rises, but the amount of oxygen required by your body is not supplied by the breathing. So তোমার যতটুক অক্সিজেন দরকার হচ্ছে ততটুক অক্সিজেন তুমি ব্রিদিং করে পাচ্ছ না. At this point your body starts to perform anaerobic respiration in addition to aerobic respiration. And soon enough your body starts to produce a lot of lactic acid. This lactic acid can be toxic, as we have already explained. So after accumulation of lactic acid, our body tries to get rid of it. Because of this, you will have noticed that when we are performing exercise, our breathing rate increase, and when we stop exercising, our breathing rate still remains very high for several minutes. Now why is that? Because আমরা exercise করতেছি, আমাদের breathing rate বাড়তেছে, ঠিক আছে, but exercise stop করার পরে still কেন breathing rate টা high থাকতেছে? Because exercise stop করার পরে তো আর আমার extra oxygen demand এর কোনো দরকার ছিল না. The reason is that that exercise period has generated a lot of lactic acid which needs to be broken down. So exercise শেষ হওয়ার পরেও আমরা ওই additional oxygenটা নিচ্ছি so that the extra lactic acid that has accumulated in our body can be broken down. This is why we continue to breathe in heavily even after the exercise is over. So here is an example. You can see SpongeBob is breathing very heavily. Why? Because he ran a long distance. Now what is the point of this? As SpongeBob performed the exercise, lactic acid built up in his system. Now in order to get rid of this extra lactic acid, Mr. SpongeBob is breathing quite heavily even after the exercise. So এই extra oxygenটা গিয়ে ওর সিস্টেমের মধ্যে যে lactic acidটা ছিল এইটাকে breakdown করবে. Now this additional oxygen which is being taken in after the exercise is called oxygen depth. So oxygen depth is the volume of oxygen that is needed to completely oxidize the lactate buildup during a period of anaerobic respiration. The next point that we'll need to understand is something called fermentation. What is fermentation? Fermentation is the process of using anaerobic respiration in microorganisms to make useful products. If you remember this equation, here you can see that microorganisms and yeast, when they anaerobically respire, the glucose gets broken down into ethanol. Now this ethanol or alcohol has a number of commercial uses, and this process of anaerobic respiration going on inside a microorganism is known as fermentation. So what are the importance of fermentation? Fermentation is used to make wine. It is used to make beer. Fermentation is also needed to make bread. Now fermentation not only produces alcohol, fermentation can produce other organic materials as well. We are going to see details into fermentation or how fermentation can be used for commercial purpose in later topics. So far we have learned a lot about respiration. But you see, the main problem with respiration is that we cannot see respiration through our eyes. Respiration is a chemical process and cannot be seen. So how do you know that respiration actually occurs? How do you know that the glucose is actually broken down? How do we know that aerobic respiration produces carbon dioxide? How do we know that fermentation produces alcohol? Since we cannot see them directly, we use some clever experiments to determine what is actually going on. although we cannot see the respiration directly, we can see the products of respiration. And we know that one product of aerobic respiration and also fermentation is carbon dioxide. Now carbon dioxide by itself is a colorless gas. However, when we pass carbon dioxide through lime water, the lime water turns milky. Here in this test tube, we have some clear lime water. Now lime water has the chemical formula calcium hydroxide, CaOH2. Now calcium hydroxide is a colorless liquid, but if we pump in some carbon dioxide, then this colorless liquid will turn turbid or milky. If we add more carbon dioxide, it is going to turn even more milky. So imagine you have some clear lime water. It suggests that the atmosphere does not have any carbon dioxide. But if you start to see that the lime water is turning milky, then you will realize, or then you can assume, that carbon dioxide is being added to it. And this is vital for the subsequent experiments. So how can we demonstrate that you are producing carbon dioxide right now? We can perform a very simple experiment. We are going to collect a test tube full of lime water, clear lime water, and if you blow into this clear lime water using a straw, you will see that the clear lime water will quickly turn cloudy. So if this was the actual color of the lime water, after blowing through it, you will see that the clear lime water will quickly turn milky. So question হচ্ছে যে কেন clear lime water টা হঠাৎ করে milky হয়ে গেল? Like we have already said, that this can only happen if carbon dioxide is added to the lime water, meaning that তুমি যখন breathing করতেছো, তোমার body থেকে carbon dioxide lime water এ যাচ্ছে and the lime water is turning milky. So this experiment conclusively prove that respiration inside our body produces carbon dioxide. Here is another experiment that can be used to demonstrate that animals or living organism produces carbon dioxide. For this, we are going to use at least 3 conical flux. In this experiment, they are using 4 conical flux. In the middle one, they have placed 2 snails. Over here, there is a conical flux containing lime water, and this one also contains lime water. And here is another conical flux containing caustic potash solution. Now, the caustic potash solution, what it does is that it can absorb carbon dioxide. So, এই সিস্টেমটার মধ্যে the air is entering through this pipe. Now, we know that atmospheric carbon dioxide contains 0.04%. Atmospheric air contains 0.04% carbon dioxide. So, we want to filter out that carbon dioxide. That's why we are passing the atmospheric air through this first chamber. So যখন atmospheric air এই chamber দিয়ে ঢুকে, atmospheric air এর যত carbon dioxide ছিল, it gets absorbed by the caustic potash. And then this filtered air enters into our first lime water chamber. So since all the carbon dioxide has been removed at this point, the air entering into chamber B does not have any carbon dioxide, and the lime water in this chamber remains transparent or clear. Then this air enters into chamber C, which contains the living organism. Now the living organisms absorb the air, and they absorb the oxygen, they respire and produces carbon dioxide. this carbon dioxide passes out through this tube and enters into the second lime water chamber. This time, since it has the carbon dioxide, the lime water turns milky. So this experiment conclusively proves that living organism produces carbon dioxide. যদি লিভিং অর্গানিজম কার্বন ডাইঅক্সাইড প্রডিউস না করত তাহলে এই মাঝখানে চেম্বার ডি would not have turned cloudy. Now lime water is not the only indicator to understand the presence of carbon dioxide. There is another very useful indicator that can be used to demonstrate the level of carbon dioxide in air, and that ingredient or that indicator is known as the hydrogen carbonate or bicarbonate indicator. Below in this picture you can see a range of different colors. Now all of them are hydrogen carbonate indicator. Normally, in normal atmospheric carbon dioxide concentration, the bicarbonate indicator takes a red or orange color. Now as the atmospheric carbon dioxide concentration increase, the red or orange bicarbonate indicator turns yellow, and if carbon dioxide level decrease, the color turns toward purple. So if you see that the bicarbonate indicator has orange color, it means the carbon dioxide level is normal. If you see that the bicarbonate solution has turned yellow, then you'll have to understand that the level of carbon dioxide has increased. And if you see that the bicarbonate solution is purple in color, this means that level of carbon dioxide is very low. Here is another experiment that we can do to prove that carbon dioxide is produced by living organism. What we are going to do is that we are going to take two test tubes. So আমরা যদি দুইটা টেস্ট টিউব নেই and দুইটাতেই bicarbonate indicator রাখি, initially দুইটা bicarbonate indicator এর কালারই থাকবে orange, because normal air এ bicarbonate indicator এর কালার থাকে orange. And then what we are going to do is that একটা টিউব আমরা খালি রাখবো, কোনো organism দিব না, আরেকটা টিউবে we are going to put an insect. We are going to seal both tubes and keep it for a given amount of time. After half an hour or after say 10 minutes, we are going to see that the bicarbonate indicator has changed its color. যেটাতে কোনো অর্গানিজম আমরা দেই নাই ওইটাতে কালার কোনো চেঞ্জ হবে না, ওইটার কালার রেড অথবা অরেঞ্জই থাকবে। However, যেটাতে আমরা bicarbonate indicator দিছি প্লাস অর্গানিজমও রাখছি, এইখানে বাইকার্বনেটের কালারটা রেড থেকে ইয়েলো হয়ে যাবে। Now red থেকে ইয়েলো হওয়া মানে কি? As we have already seen, যে রেড থেকে ইয়েলো হয়ে যাওয়ার মানে হচ্ছে যে ওই চেম্বারে কার্বন ডাইঅক্সাইড লেভেল ইনক্রিজ করছে। So this experiment conclusively proved that living organisms produces carbon dioxide, and this carbon dioxide is actually produced by respiration. Now both these experiments, this one and the other one, this one, they can be carried out by insects, by small organisms like snails or frogs, also by germinating seeds. All of them will produce same results, but for faster results it's better to use some sort of animals like snails or insects because they have higher rate of respiration. Plant seeds will also provide the same result, but it will take longer because respiration in plant is slower than respiration in animal. However, while performing this type of experiments, we'll have to be very careful because we are using living organisms. although they are small organisms or although they are insects, but still they have life and we need to respect the life. So we have to be cautious about the ethics of our experiment. We should not harm the organisms during this experiment. For example, in this experiment, if we keep the insect trapped for 1 hour, then what will happen? The insect will starve of oxygen and eventually একটা সময় দেখা যাবে ইনসেক্টটা ট্র্যাপ থাকতে থাকতে মরে গেছে. So that's why we have to perform this experiment very rapidly. যতটুক সময় দরকার কালার চেঞ্জ অবজার্ভ করার জন্য আমরা ঠিক ততটুক টাইমই রাখবো. If it's done in 5 minutes, then fine. 5 মিনিট পরে আমরা ইনসেক্টটাকে ছেড়ে দেব. 10 minutes পরে আমরা ইনসেক্টটাকে ছেড়ে দেব. But আমরা এটাকে বেশিক্ষণ আটকায় রাখবো না, because if we lock it up for long period of time during lack of oxygen and food, this insect might die. Same applies for the snails. এই স্নেইলগুলোকে আমরা এখানে বেশিক্ষণ ধরে লক করে রাখবো না, because আমরা যদি এটাকে বেশিক্ষণ এখানে রেখে দেই, the organisms might die. Another thing about this experiment is that look at the orientation of the pipes. If you look at this pipe, you see এইটার beginningটা it's above the level of lime water, and এই pipeটা it is dipped in the lime water. We have to arrange these pipes very carefully, because এই pipeটা যদি it was dipped in water, তাহলে কিন্তু এখানে কোনো এয়ার ঢুকতো না এই পাইপ দিয়ে. So we have to be very careful যে first pipeটা it should be above the level of liquid, and the second pipe it should be dipped inside the liquid. Here is another experiment. This experiment will demonstrate that oxygen is used up during respiration. Here in this experiment, we are placing an insect inside a conical flux. This is an airtight chamber. However, there is a delivery tube or delivery pipe attached to the apparatus. There is also some potassium hydroxide. And over here you can see that inside this capillary tube there is a drop of liquid, and below it there is a scale. Now এই apparatusটা আমরা যদি এইভাবে সেট করে if we observe for 5 minutes or 10 minutes, you will notice that as the time passes on, this liquid starts to move in this direction. This experiment proves that oxygen is used up during respiration. So কি করে আমরা একটা মুভিং ড্রপলেট দেখে বুঝতে পারতেছি যে এখানে অক্সিজেন অ্যাবজর্ব হয়ে যাচ্ছে? It's very simple. You see, a apparatus, it is a closed chamber, right? So air can neither come in or go out. Air যদি পাস করতে হয় তাহলে it has to pass along this line. এটা ছাড়া আর কোনো ওপেন চ্যানেল নাই এখানে. So আমরা যখন এই ইনসেক্টটাকে এখানে রেখে দিচ্ছি, insecta it's going to absorb oxygen from the conical flux and release carbon dioxide. However, carbon dioxide টা কিন্তু আর conical flux এ থাকবে না, rather carbon dioxide টা potassium hydroxide absorb করে নিবে, আর অক্সিজেনটা ইনসেক্টে নিজে কনজিউম করে নিচ্ছে. So this is going to create a partial vacuum. This vacuum will draw in air from this part of the tube. যেহেতু এখান থেকে এয়ারটা এখন সরে গিয়ে conical flux-এ চলে আসছে, this will pull the colored liquid towards the conical flux, and this proves that oxygen is taken in by the insect. এই লিকুইডটা যত তাড়াতাড়ি মুভ করবে তার মানে এই ইনসেক্টটা তত বেশি অক্সিজেন নিচ্ছে. লিকুইডটা যদি আস্তে আস্তে মুভ করে তার মানে ইনসেক্টটা অল্প অক্সিজেন নিচ্ছে. লিকুইডটা যদি তাড়াতাড়ি মুভ করে তার মানে ইনসেক্টটা বেশি করে অক্সিজেন নিচ্ছে. আর অক্সিজেনটা কেন নিচ্ছে? obviously রেসপিরেশনের জন্য. So this experiment demonstrates that oxygen is used up during respiration. Now, In the equation for respiration, we have shown that respiration produces energy. But how do we know that respiration produces energy? We can prove that energy is produced during respiration by this experiment. For this experiment, we are going to take 100 germinating seed. Now germinating seeds are growing seeds. That means they are going to perform a lot of respiration. So we actually germinating seeds, we are going to divide it into two groups, 50-50. 50 of these seeds will be boiled, আর বাকি 50 seeds will not be boiled. Now যে seedগুলা, যে 50 seeds were boiled, they have died. So these are dead seeds. And the 50 seeds that were not boiled are still living seeds. So dead seed কে আমরা একটা vacuum flask এর মধ্যে রাখবো, আর living seed কে আমরা আরেকটা vacuum flask এর মধ্যে রাখবো. And we are going to insert thermometers in each of these flask. And we are going to seal the top of the vacuum flask with cotton wool. Now vacuum flask is an insulator. So ভিতরে যদি কোনো temperature change হয়, এই টেম্পারেচারটা ভিতরেই থাকবে, and this will be recorded by the thermometers. So apparatusটা এইভাবে করে সেট করে we are going to leave it for 30 minutes. Now 30 minutes later, what we will see is that if the experiment has been done correctly, the thermometer that was placed in living seed will show a greater temperature than the thermometers placed in boiled seed or dead seeds. কেন? Because living seeds যারা ছিল, they were performing respiration and they were producing energy. This energy increased the temperature inside the vacuum flask, and this temperature was recorded by the thermometer. But এইখানে যেহেতু these are boiled seed, so the seeds were already dead and they were not carrying out any respiration, and the temperature inside the vacuum flask remained the same. এই এক্সপেরিমেন্টটা করার জন্য আমাদের সিডগুলোকে একটা disinfectant solution দিয়ে ওয়াশ করে নিতে হবে to prevent the growth of the bacteria. আমরা যদি disinfectant দিয়ে সিডগুলোকে ওয়াশ না করি, so disinfectant হচ্ছে অ্যান্টিসেপটিক বলাটা বেটার. সো আমরা যদি অ্যান্টিসেপটিক দিয়ে এটাকে না ওয়াশ করি, তাহলে যেটা হবে যে ওদের ভেতরে যেই ব্যাকটেরিয়াগুলো আছে, এই ব্যাকটেরিয়াগুলো সিডকে ডিকম্পোজ করে ওরা আবার একটা আলাদা টেম্পারেচার দিতে পারে. So that will confuse the results. So better to wash them with antiseptic solutions. Another point we mentioned about respiration was that we said glucose is broken down. Now how can we demonstrate that respiration breaks down glucose? We can understand that the glucose is broken down by this experiment. For this experiment, we are going to take 100 germinating seeds, and again we are going to divide them into two groups. 50টাকে we are going to boil, and 50 will be left unboiled. And then we are going to place these seeds separately on moist cotton wool for 1 week. Idea is that এই 1 week এর মধ্যে the unboiled seeds will continue respiration and 50 boiled seeds will not continue respiration. And after 1 week, we are going to heat the seeds in oven and measure their dry mass. It will turn out that unboiled seed will have a lower mass than the boiled seed. কারণ কি? Because unboiled seeds were alive. So they carried out respiration and they broke down their glucose, and as the glucose was broke down and carbon dioxide and waters were released, they escaped into the atmosphere, so ওদের ওয়েটটা আস্তে আস্তে কমে গেছে. But boiled seeds, since they were dead, all the glucose remained stored in their body. So যেহেতু ওদের গ্লুকোজের লেভেলে কোনো চেঞ্জ হয় নাই, ব্রেকডাউন হয় নাই, that's why ওদের mass ওদের চেয়ে comparatively বেশি ছিল. Now let's come to fermentation. We said anaerobic respiration in microorganisms or fermentation produces carbon dioxide. How do you know that carbon dioxide is produced by anaerobic respiration? We can demonstrate that carbon dioxide is released by fermentation using this setup. In this experiment, we are going to use two test tubes. In one test tube, we are going to place some glucose solution with yeast suspension, and in the other test tube, we are going to place some clear lime water, and the two tubes will be collected using a delivery tube. You can see that there will also be a layer of oil on top of the glucose solution. Now this setup, if we leave it for say 30 minutes, then after 30 minutes we will see that the colorless lime water will turn turbid or milky. The question is, why did this happen and what does this demonstrate? Now আমরা আগেই বলছি যে when we see that clear lime water is turning milky, it only means that carbon dioxide is being added to that solution. তাহলে এই carbon dioxide টা কোথা থেকে আসলো? Obviously it has come from this delivery tube, and this delivery tube got its carbon dioxide from this tube. So এই tube কোথা থেকে carbon dioxide আসলো? It came from the yeast. The yeast is a microorganism and it was in the glucose suspension. So the yeast, what they did was that they broke down the glucose by fermentation, and as a byproduct of that breakdown, carbon dioxide was produced, and this carbon dioxide entered the delivery tube and reached the lime water and turned the clear lime water milky. Now the question is, why are we using a layer of oil on top of the glucose solution? The reason is that yeast is a microorganism that can carry out both aerobic respiration and anaerobic respiration. So yeasটের কাছে যদি অক্সিজেন available থাকে ও aerobically respire করবে, but অক্সিজেন যদি না থাকে তাহলে anaerobic respiration করবে. Now we want to demonstrate that yeast is carrying out anaerobic respiration. তাহলে we have to get rid of all the oxygen from this system. কিন্তু আমরা যদি টেস্ট টিউবটা এইভাবে রেখে দেই, এই যে এয়ারটা এখানে তো ঠিকই অক্সিজেন আছে, এই অক্সিজেনটা যদি এখানে মিশে যায় তাহলে কি আর ফার্মেন্টেশন হবে? হবে না. So in order to separate this oxygen from mixing with the yeast, we have placed a layer of oil on top. We are almost at the end of this lecture. Now before ending, let's talk about ATP. Now ATP is this. Its full form is adenosine triphosphate. It is a molecule that has two phosphate heads attached to it. Now this ATP is an unstable compound, meaning ATP যে কোনো সময় ভেঙে যেতে পারে. ATP যদি ভেঙে যায়, it produces another compound called ADP or adenosine diphosphate. The reason it's called diphosphate is because এটার সাথে দুইটা ফসফেট লাগানো থাকে. তাহলে এটাতে তিনটা ফসফেট ছিল, এটা যখন ভেঙে গেল, it formed ADP and a single phosphate molecule. Now এই রিঅ্যাকশনটা রিভার্সিবল. ATP ভেঙে ADP আর ফসফেট হতে পারে. Again, ADP and phosphate can recombine to form ATP. When ATP breaks down to give ADP and phosphate, it releases energy. And when we give energy, or when there is an input of energy, the ADP and phosphate joins together to reform the ATP. তাহলে ATP ভাঙলে এনার্জি পাবো, আর এনার্জি দিলে ADP আর ফসফেট কম্বাইন করে ATP পাবো. Now here in these equations we have seen যে যখন respiration হয়, গ্লুকোজ ব্রেকডাউন করে আর এখানে এনার্জি প্রডিউস হয়. কিন্তু এই যে এনার্জিটা রিলিজ হচ্ছে, this energy cannot be utilized by body cells. Rather, রেসপিরেশন থেকে যে এনার্জিটা রিলিজ হয়েছিল ওই এনার্জিটা চলে যায় এটিপি মলিকিউলসের মধ্যে। তাহলে গ্লুকোজ breaks down, energy is produced, and that energy is converted into ATP. One molecule of glucose produces enough energy to make 38 molecules of ATP. This is when the respiration is occurring aerobically. And we say যে anaerobic respiration এ কম energy produce হয় because যখন anaerobically respiration হয় one molecule of glucose produces only 2 molecules of ATP. Now the reason ATP is produced is because ATP আমরা অনেক manageable ফর্মে ইউজ করতে পারি। ডাইরেক্টলি রেসপিরেশন থেকে যে এনার্জিটা হচ্ছে এই এনার্জিটা এটিপি ছাড়া অন্য কোনো ফর্মে কিন্তু স্টোর করা পসিবল না, অ্যান্ড একটা গ্লুকোজ ব্রেকডাউন করলে এত বেশি এনার্জি হয় that energy cannot be utilized by the cell. So that mass of energy is stored into smaller chunks in the ATP molecules. So it is more like this. ধরো তোমার কাছে তুমি একটা দোকানে গেছো and you want to buy a pen and the pen cost 80 টাকা, sorry 8 টাকা, but the cashier does not have any change. তোমার কাছে 4টা নোট আছে, you have a 30 টাকা note. I know the 30 টাকা note doesn't exist. Just for the sake of this experiment, let's think that you have a 30 টাকা note and you also have 3 10 টাকা notes. So 30 টাকা and 3 10 টাকা দুইটার ভ্যালু কিন্তু actually same. Now if you give this 30 টাকা to the cashier, সে তোমাকে কোনো কিছু রিটার্ন দিতে পারবে না। পেনটার দাম ছিল 8 টাকা, so তোমার বাকি 22 টাকা will be wasted. So তুমি কি ক্যাশিয়ারকে 30 টাকা দিবা নাকি তুমি ক্যাশিয়ারকে 10 টাকা দিবা? Because তুমি যদি ওকে 10 টাকা দাও, you are going to get the pen and তোমার লস হচ্ছে just 2 টাকা. So obviously you are going to give him 10 টাকা. Now the same principle can be applied to understand the concept of glucose and ATP. আমি যদি ডাইরেক্ট আমার সেল যদি ডাইরেক্ট গ্লুকোজটাকে ব্রেকডাউন করে সব এনার্জি দিয়ে দিত, তাহলে ওর একটা অ্যাক্টিভিটির জন্য হয়তো দরকার ছিল 8 জুলস এনার্জি, বাট টোটাল গ্লুকোজটা ব্রেকডাউন করে ফেললে যেটা হতো যে 30 জুলস এনার্জি বের হয়ে যেত, 22 জুলস ওয়েস্টেড হয়ে যেত। ইনস্টেড আমার সেল যেটা করে যে ও ও গ্লুকোজ মলিকিউল ভেঙে যে এনার্জিটা পাচ্ছে ওই এনার্জিটা ও রিলিজ করে না, রাদার ওই এনার্জি দিয়ে ও এটিপি মলিকিউল বানায় নেয়। না কথার কথা একটা গ্লুকোজ মলিকিউল 30 জুল এনার্জি দেয়, ওই সেম গ্লুকোজ দিয়ে আমি 3টা এটিপি বানায় নিতে পারি। each of these ATP will now give me 10 joules of energy. So next time আমার যখন ওই 8 joule activity-টা দরকার হবে, I am not going to use this entire glucose molecule, rather I am going to use one of the ATP molecules. তাহলে যেটা হবে যে only এবার আমার 2 joule energy waste হচ্ছে। So this is why we are using ATP, and ATP can utilize energy in more manageable form. So the lesson here is that the food from or the energy from the food is first converted into ATP, and this ATP is broken down to release the energy for cellular work. ATP gets converted into ADP and phosphate, and next time আবার যে আমরা respiration করি ওই এনার্জি দিয়ে এই ADP phosphate combine করে আমরা আবার নতুন ATP বানায় স্টোর করে রাখি। So that's it for this class. Thank you all. See you in the next class.